We're going to talk about enzyme mode of action featuring Barry the whale. The first question we have to ask is, what is an enzyme? Pretty much an enzyme is this big old honky protein with many different amino acids all bound together by hydrogen bonds, covalent bonds, ionic bonds, hydrophobic interactions, van der Waals forces. There's a ton of them. But the main three parts we're going to look at is the enzyme itself, aka Barry, the substrate, which eventually binds to the enzyme, and the active site, for which the substrate, of where the substrate binds to the enzyme. So why do we need enzymes? Well, pretty much every single human body function we have requires proteins to operate, such as digestion, respiration, muscle function. And with these enzymes, these functions are able to occur within milliseconds, almost instantaneously. Now, if we take the enzymes away, these functions will take so long, we won't be able to live no more. They just take so long that we deteriorate before anything even occurred. Now, let's look at those enzymes and the energy curve associated with them. So let's take a trip, for example. You want to go visit your grandma, who lives in Edmonton, and you live in Prince George. To do that, you have to fly all the way over the Rocky Mountains. They're right in between. To start with, this is the main path you would have to take. So you're going to hop in your airplane, your little crop duster from Prince George, and you're going to fly all the way over the Rockies and land in Edmonton. This is going to be the basic reaction. This would be the reaction with no enzyme. And this curve, from the bottom of the curve, top of the curve, to the bottom, this is called the activation energy, also known as the energy needed for the reaction to occur, to take the substrate, you in Prince George, and create it to a product, you in Edmonton. Now, let's get an enzyme involved. So the red line shows the enzyme curve when an enzyme is involved. So let's take a jet now. Think of it as the jet is a super version of the reaction with the enzyme. That this jet is able to go right through the mountain instead of over top of it because it flies so fast. So you hop in a jet, you fly straight through the mountain, and you land in Edmonton. Now this reaction is the one with the enzyme. And this reaction has a reduced activation energy because the energy needed for the reaction to take you to Prince George to you in Edmonton, or you as a substrate, and transfer you to a product is less, therefore it's reduced. Now let's get into the nitty-gritty of how this occurs. How do then enzymes operate? So let's take Barry again. Barry's going to come back in our example. And Barry's got his two backpacks, substrate 1 and substrate 2. Now these substrates are going to come close to the enzyme. They're going to form those non-covalent bonds with the substrate. Now we get Barry 2.0, because what happens is when these substrate bind to the enzyme, they're going to change the conformation of the enzyme so they fit super nice and snug, and they can hold the substrate super well. This is what we call the induced fit model. Continuing, we get Barry once again with his two substrates, but what we notice is that these two substrates are slowly turning into product. This is what we call the transient state, or also known as the transition state. It's the most high energy part or state of this enzyme substrate complex, and this with the most going on. And now, once all of the substrates have been turned into product, the enzyme eventually will release these products. They will go and do whatever they need to do, and the enzyme will convert back to its original conformation, ready to get more substrates and redo the whole process once again. But how do enzymes exactly lower the activation? We know how they bind together and we know how they operate and change conformation, but how do they lower the activation energy? Well, we're going to take Barry again in that very first state when he has his two substrates and they bound with the enzyme, forming those non-covalent bonds. So we talked about these bonds could be hydrogen bonds, they could be hydrophobic interactions, they could be van der Waals forces, or even ionic bonds. There's a bunch of them going on in there. But what happens is with all these interactions, they release a little bit of binding energy when they form, when they break, when everything's going on in there. And this binding energy, when enough of it conglomerates all together, is enough to lower that same curve we talked about and actually make that reaction occur so much quicker.